Okay? Does that help?
Yes.
Okay. That's good. So this is me, you know who I am. This is my 44th year at Howard. 44, November 1 was 44 years, so I'm in my 45th year. Even though I'm emeritus, which means I'm retired. I'm not on Howard's salary, but I do what I want, when I want, and how I want.
Oh, period.
So I'm retired. So look forward to it.
But are they still paying you though?
I've got retirement, but Howard's not paying me anymore. My retirement's off with Citibank somewhere in New York or something.
You doing this for free?
I'm doing this for free because I love you guys.
Okay.
It's because Dr. Jackson and Dr. Marshall showed me a great deal of respect when I was here, so I've offered to give them some guest lectures, okay?
Thank you.
This is free, guys. None of your tuition money's going to me, all right? Okay, it's okay. It's all right. I'm ready to go.
That's a difference.
So let's have some fun. Early development. So you recognize this is one of the first slides we had. And what's this? This is the uterus over here, right? Fallopian tube, fimbriae, and the ovaries out here, which expels eggs. And they are drawn into the fallopian tube by these fimbriae. And after one day, you have division. What kind of division is this? Is it meiotic division or mitotic division? Mitosis, right? It's cell duplication. And one day at a time, you get duplication of these cells. So, day 2, you have a 4-cell stage. Day 3, you have an 8-cell stage. Day 4, you have a 16-cell stage, morula. And by that time, the next day, day 5, you have a 16-cell stage. But what's happening? They're not filling the entire center, but they are concentrated at one end of the blastocyst, right? You recall what we call this? It's called a pole, embryonic pole, right? And the other side of the cell is called the ab-embryonic pole, away from. Ab is Latin for away. And so after about day 5, it gets expelled from the fallopian tube, and it goes through a process called hatching where the outer membranes are lost. And this blastocyst is looking to implant in the uterine wall, right? All old news for you. So, you look here, here's the uterus, and here's the walls of the uterus. There are 4 walls, one each side laterally, one anterior, one posterior, and then superiorly. And if the blastocyst implants on one of these walls, it's a normal pregnancy beginning. However, if it implants in the fallopian tube, or even on the posterior wall of the abdomen, or down here in the cervix, it is an abnormal implantation site, and the pregnancy is at risk. It's in the fallopian tube, so the fallopian tube can burst from the embryo developing, right? And that can be fatal to the mother. So here shows you the uterine wall. Here's the blastocyst. Here's the embryonic pole. And we have here the cytotrophoblast. These appendages coming off the embryonic pole are called the syncytiotrophoblasts, and they're labeled here, okay? Syncytiotrophoblasts are these appendages that develop at the embryonic pole. These normal cells here are cytotrophoblasts, and they're surrounding a blastocyst cavity. And we see early on developing, day 7, end of the first week, we see a bilaminar disc. Bi means 2. Lamina means layers, 2-layer disc. And you see here, these yellow cells are called hypoblast, and the blue cells are called epiblast. It's a 2-layered cell, or bilaminar, that means 2-layer disc. So here is the blastocyst cavity. And what we're going to show in the second week is a lot of things going on in transformation of this bilaminar disc. If you need me to repeat anything, you can stop me. We have plenty of time. So, you're okay? I'll keep going. So here, day 8, the beginning, first day of the second week, you see this blastocyst with its bilaminar disc embedding in the uterine wall. This is accomplished by the syncytial trophoblast, these appendages, which facilitate bearing the blastocyst into the uterine wall. And here, now you see something going on here. This blue area of the epiblast is undergoing some transformation. And you have an opening up here at the embryonic pole of the amniotic cavity. The amniotic cavity is associated with the epiblast. Okay? Hypoblast is here. It's not undergoing any transformation yet. These are capillaries in the maternal uterine wall. Cytotrophoblast, blastocyst cavity. So by day 9, the second day of the second week, you see that the bilaminar disc and the whole blastocyst is buried completely in the uterine wall. It closes off at the abembryonic wall with something called the coagulation plug. Now, you see that the amniotic cavity is opening up more. There are cells called amnioblast cells that are facilitating this opening. The hypoblast here undergoes a transformation on the lateral edges. Of the hypoblast, you have very rapid cell division. And these cells are going to migrate here on the interior side of the cytotrophoblast. And they form a structure called Heuser's membrane. Heuser's membrane is also called the primary yolk sac. They're synonyms. Okay. So you have a blastocyst cavity here. You have a cytotrophoblast. You have a syncytial trophoblast. You have a hypoblast, yellow, epiblast in blue, amniotic cavity, and here you have this blastocyst cavity. Now this is a little bit complicated, so I'll make sure that you get it, all right? So here is an implanted bilaminar disc. It's within the uterine wall. You have cytotrophoblast, these cells here. You have the syncytial trophoblast that is dispersed within the uterine wall. You have the blue cells, which are epiblast, the yellow cells, which are hypoblast, and here, associated with the epiblast, you have the amniotic cavity. What's going on here is that there is, we've had really rapid cell division to make the primary yolk sac, or Heuser's membrane. But you have appearing here a structure called the extra-embryonic reticulum. This is a acellular matrix that resides between Heuser's membrane and the cytotrophoblast. Fills this potential space between the primary yolk sac, or Heuser's membrane, and the cytotrophoblast. This pink substance here, it's like styrofoam. No cells involved. What happens though is it forms a template by which these cells that are dividing here on the edge of the epiblast, on both sides of the edge of the epiblast, these cells form two layers. One layer is between the extra-embryonic reticulum and Heuser's membrane, right there. And another layer is between the cytotrophoblast and the extra-embryonic reticulum. So 2 layers. So the function of this extra-embryonic reticulum is to form a template that will guide these 2 layers to their disposition. Why do we call it extra-embryonic reticulum? Excuse me, extra-embryonic mesoderm. Why do we call it extra-embryonic mesoderm, these 2 layers? It's outside the embryo. Whereas the mesoderm on the embryo going to develop, it's going to develop in the beginning of the 3rd week here between the epiblast and the hypoblast. This is outside. And it has its own fate, which we will discuss. So this is a very important process. There's 2 layers of extra-embryonic reticulum at day 12. And the next process that occurs is that, once again, the lateral edges of the hypoblast have cells that undergo very rapid multiplication, and they migrate down here on the interior of the mesoderm, extra-embryonic mesoderm. And what they do is they push out the primary yolk sac here progressively until they become these remnants of the primary yolk sac. And this is going to be at the abembryonic pole. This is the embryonic pole. So now you have a bilaminar disc, epiblast and hypoblast. You have an amniotic cavity, and you have this other cavity here, and you have the dangling in this area of the chorionic cavity. This is the chorionic cavity. So yolk sac, amniotic cavity, bilaminar disc, epiblast and hypoblast. And this shows you the fate at day 15. Forget this 15 days. 15 days is the beginning of the 3rd week. So you have chorionic cavity here. You have the extra-embryonic mesoderm here. This is the embryonic pole. You have a bilaminar disc, epiblast and hypoblast. Associated with the epiblast is the amniotic cavity. Associated with the hypoblast is the yolk sac, the definitive yolk sac. And here at the abembryonic pole on the other side is the remnants of the primary yolk sac. Did you all get that? Is there anything I need to go over again that you didn't get? Yes, ma'am.
The Hauser's membrane, you said about just the yolk sac?
Hauser's membrane is the primary yolk sac. It's the first one that's generated from the edges of the hypoblast. The secondary yolk sac, which comes later, is called the definitive yolk sac.
The what?
Definitive yolk sac. Definitive. So there's 2 phases of yolk sacs. There's one phase of extra-embryonic mesoderm, and we're going to go into the 3rd week, and we're going to develop intra-embryonic mesoderm. So this is the end of the pre-embryonic period. So now that you know this, what do you think about IVF, and what do you suppose is the likely stage at which these eggs, fertilized eggs, are harvested and implanted in the womb? At what stage would you expect them to be? Well, there'd be a stage of development before implantation, right? So if we implant the embryo beginning around day 7, is this an embryo? The embryo. So what's all this discussion about destroying embryos? They're not embryos yet. Okay? They're not embryos. They've not been implanted, right, in the uterus, and they haven't developed through a bilaminar disc stage, or trilaminar disc stage, or anything. So I'm amazed at all this conversation we had during the past election about embryos, destroying embryos. They're not embryos. And you're a scientist, you're a young scientist, you're trained in biology, physiology, anatomy, all these different basic sciences you have. You should be aware of what they're talking about, right? Now, I don't have any message about IVF or abortion or whatever. I just want you to be informed about what the likely stage of development we have when we implant cells in the uterine wall of a woman, and they develop from there. They're in the pre-embryonic stage. Most certainly have to be pre-embryonic stage. Because bilaminar disc and trilaminar disc have to be developed within the uterus. Right? Does that make sense to you? I haven't studied IVF, I haven't studied, the process or anything like that, but I'm just trying to use logic here. Any questions? Yes.
On picture C, you were saying one end is the embryonic pole and one end is the embryonic pole. Can you
This is
go
where
to
the embryo develops, is at the embryonic pole.
that? Okay. And then, okay.
This whole mass is at the embryonic pole. The one that's in contact with the embryonic pole is the epiblast.
Okay. Thank you.
The embryonic pole is at the other side, and that's where those remnants of the yolk sac are isolated. So we're going to find out more about what the fate of the epiblast is and what the fate of the hypoblast is in the next slide. So, this is day 15. This is the beginning of the embryonic period. The embryonic period is from week 3 through week 8. So day 15 is the first day of the third week. Okay? So what we have here is the extraembryonic mesoderm on the outside. Syncytial trophoblast deep to that. You have a bilaminar disc, which means you have an epiblast here, and below it, the hypoblast. Amniotic cavity is associated with the epiblast. Yolk sac is associated with the hypoblast. And you know that you're at the beginning of the embryonic period because this structure appears on the surface of the epiblast. Okay? So it's called the primitive streak in its entirety. And you have a primitive node. This is the cranial end, or the rostral end. This is the caudal end, or the tail end. So you have a primitive node, swelling. Within the center of the node, there's a primitive pit. And extending towards the tail end is a primitive groove. These are all structures on this surface which are epiblast, right? They're on the epiblast side. You see these two membranes, bucopharyngeal membrane and the cloacal membrane. We're going to talk about the development of intraembryonic mesoderm, which is going to develop along this streak and fill a potential space between the epiblast and the hypoblast. The bucopharyngeal membrane and the cloacal membrane have no mesoderm migrating into these areas because this is going to be the opening of the oral cavity. And this is in a bird, for instance, which don't separate the anal canal from the birth canal. They have a cloaca. This is open at the, becomes perforated and open at the other end. So, we have a bisected bilaminar disc. We have a primitive streak here. Here you have a primitive node, primitive pit, primitive streak. It's been hemisected. You can see here in blue the epiblast, in yellow the hypoblast. And what you have is a process called gastrulation. gastrulation and development of a 3-layer disc or trilaminar disc. So this next slide shows you the process of gastrulation, development of the trilaminar disc. Now you see these cells are dividing along the primitive streak, and they're rapidly migrating towards the hypoblast, and they're replacing hypoblastic cells to form the endoderm germ layer. Endoderm. So once the endoderm is formed, definitive endoderm, that's the final endoderm, you have cell division continuing along the primitive streak, and you have another germ layer developing in this potential space, we call it potential space between the epiblast and the endoderm. And this is the middle germ layer. Meso means middle, mesoderm. It's not extraembryonic mesoderm, it's intraembryonic mesoderm. Okay. So here you have a coronal section through the developing embryo. You have here amniotic cavity associated with a layer of epiblast. Yolk sac associated with hypoblast. So now we become endoderm. And we have mesoderm. Once mesoderm is fully formed, epiblast becomes ectoderm. So you'll have a trilaminar disc, ectoderm, mesoderm, and endoderm. Recall that epiblast is associated with the amniotic cavity. Endoderm is associated with the yolk sac. This is a fate map. Okay, this is a fate map of the development of mesoderm and its migration in this potential space between the ectoderm and endoderm. So you see the mesoderm that's formed around the primitive node, the primitive pit, migrates directly to rostrum or towards the cranium. Rostrum means snout. So towards the cranial end. Remember this mucopharyngeal membrane. Focal membrane is here. So initially there's migration anterior, then laterally, and essentially, eventually posterior. This primitive streak grows in its length as the embryo develops. So here, day 17, again, ectoderm in blue, amniotic cavity, superior to it. Endoderm in yellow, yolk sac below it. And now we have the intraembryonic mesoderm differentiating. Towards the axis, you have this population of cells here called paraxial mesoderm. That means on either side of the axis, paraxial mesoderm. Lateral, you have here intermediate mesoderm, and further laterally is the lateral plate mesoderm. So the lateral plate mesoderm develops into 2 layers, one associated with the amniotic cavity, the other associated with the yolk sac. The one associated with the amniotic cavity is the somatophoric mesoderm. The one associated with the yolk sac is called the splanchnophoric mesoderm. Somatophoric mesoderm lines the interior of the body wall, the ceiling. Thoracic cavity, abdominal cavity, and pelvic cavity. It's the lining. It's the lining. Splanchnophoric mesoderm, we'll see in another slide, is going to invest the alimentary canal. Okay, we'll see that shortly. This is another view, another view here of a cutaway, paraxial mesoderm along the axis next to the notochord process here. Then there's intermediate mesoderm, and then lateral plate mesoderm. So paraxial mesoderm is going to be responsible for things like your spinal column and associated muscles. Okay? Intermediate mesoderm is going to be committed to your general system. Lateral plate mesoderm is going to develop limbs, for instance. So here we go through a series of slices. You see here paraxial mesoderm. This is your neural canal here, which developed from the overlying ectoderm. You have aorta. You have two dorsal aorta. And you see them in a single one. And then a notochord process on the notochord. So what's going on here? These structures are called somites. Somites. You'll recall all somites are preceded by somitomeres. The first 7 somitomeres never develop into somites. All the rest of the somitomeres down the column, spinal column, form somites. So here we see for a typical somite, the medial and ventral walls of the somite break down, and these specialized cells called sclerotome migrate and invest the neural tube. And you can see they also invest the notochord. So here is a presumptive vertebral body, neural tube, the body, and you get the lamina. Okay, you have all the structures in a precartilaginous form of a vertebrate. So, presumptive vertebral body, and you see that the notochord becomes embedded here within the body of the vertebral body. Out here you have a structure called the dermal myotome that differentiates into a myotome, which is a deep muscle mass here, and a dermatome. What's going on here is this is going to be your lateral plate mesoderm, right here, this block. And there's 2 parts of this that differentiate from the lateral plate mesoderm. You have what? The somatic mesoderm and the splanchnic mesoderm. Somatic mesoderm is going to line the body wall, which you see here. The splanchnic part of mesoderm is going to line the gut tube. So the splanchnic part of mesoderm is very interesting because it becomes muscles of the gut and cartilages of the gut and fibrous tissue of the gut. Where do you have cartilage and fibrous tissue in the gut tube? Where do you have it?
Caloric sphincter.
You have it in the lungs, right? Lungs are outgrowths of the endoderm. And they get invaded by mesoderm from the gut tube that form cartilages of the lungs, fibrous tissue, etc. You don't have those kinds of tissues below the diaphragm. So, splanchnic lateral plate mesoderm, somatopark, lining the body wall. That's the fibrous tissue you see inside, for instance, the thoracic cavity. And then the splanchnic mesoderm here. It's lining the gut tube. So what kind of tissues do you have in the gut tube that are mesoderm derivatives? Time to think about that. What? So go through peristalsis, what do you have to have? Muscles. Yeah. Are these muscles under voluntary control or involuntary control? Involuntary control. That's right. You got it. So there's muscular tissue that's directly associated with the lining of the gut. What is the germ layer that forms the lining of the gut? Mesoderm, ectoderm, or endoderm? Endoderm. That yellow stuff that we see, the yellow tube, that represents the endodermal lining of the gut. It's from the most anterior part of the oral cavity up until the anus, up through the rectum. Endoderm. And it's from that lining. And that lining is a very sensitive lining. It's very sensitive. In sort of final stages of life, one determinant of death is that the endodermal lining breaks down and becomes incapable of its function. And you just start bleeding from the anus or the oral cavity. Any questions about this so far? Is there anything that's unclear? Okay, that's good. You should define all my questions. You know all this, you define. More importantly, you should do it all on your boards. So here, we have the presumptive vertebral body, the neural tube, aorta. There's a body cavity, somatopleuric mesodermal lining, splanchnopleuric mesodermal lining around the gut tube. And now we're going out here to lateral structures, dermomyotome. Dermomyotome has two layers. One is the dermatome, which is the deep skin component. The other one is the myotome. And what's important here is the myotome differentiates into a dorsal epimere and a lateral hypomere. So, what primary rami innervate the epimere? Dorsal primary rami. That should be laser etched in your brain. Okay, after all the time we spent on ventral primary rami and dorsal primary rami, so the hypomere here is representing muscles of the appendicular skeleton, the body wall, both anterior, laterally, and posteriorly, and then the superficial intermediate muscles in the back. The epimere forms the deep musculature of the back, innervated by dorsal primary rami. All the hypomere muscles are going to be innervated by ventral primary rami. Okay? Is that laser etched into your brain? It's a very important principle for us. So these rectus column muscles, these hypaxilla muscles, which are in the ribs and the abdominal wall, these are all ventral primary rami. And something very important, and I expect you'll have questions on this on your boards, because 90% of all NIH dental research is on this. Okay? The fate of neural crest cells. The fate of neural crest cells are important because there's a lot of deformities of the face, facial clefts, for instance, and other associated disorders that are due to the fate of neural crest cells. So, here is the neural crest. This is the ectoderm, and ectoderm undergoes some folding, and then downward projection on either side of the mesoderm to form a neural tube, which is here. So this folds, closes off on the top, and drops down to form a neural tube. Okay. Dorsal system, notochord. So we have here paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. So you have somatophoric mesoderm and spicular mesoderm, it's all over here. So what happens is at this crust, there is something like gastrulation, but it's neural crest cells undergoing rapid division. And these cells migrate throughout the body. And much of the research is how are they signaled to go to where they end up? How are they signaled to do that? What determines where they go to form the structure they do? And is there determination here at this point? Or is there signaling on the way that causes them to become structures that are their fate? And this gives you some ideas. Melanocytes, or skin color. Dorsal root ganglia, chain ganglia, pre-aortic ganglia, and enteric ganglia. Do any of you recall what the enteric system is? Enteric nervous system, it's its own nervous system. It's for the gut. There's millions upon millions of cells that regulate gut functions. And this is very poorly understood, how this works. But it's independent of your spinal nerves. It's its own nervous system. That's all I have to say about that. So, it's guaranteed you'll have at least one question on this table. Okay? So, multiple choice, right, is the kind of question. I have 4 of these that are derivatives of neural crest cells and 1 that's not. It should be obvious to you. But what's important for you is odontoblasts, pharyngeal arches, cranial bones, and in neuro, Schwann cells, glial cells, we said melanocytes, enteric ganglion, etc. Adrenal medulla, dorsal root ganglia, chain ganglia. I can ask 4 of these that belong and another one that doesn't belong. I usually do that. Familiarize yourself with this because this is at the center of dental research. So, somebody on the committee is going to ask you about something relevant to dental research, and this is the likely set of questions. So, do you want to take a break? Before we go to break, John, would you like a break? Somebody say yes. I'd like to have a break. 5 minutes.